In MCQ 8, we're dealing with the organ, liver. This is a very iconic question together with enzyme in O Level. I will always start out this question by labeling out the different kind of vessels. The artery that's feeding into the liver itself, it will actually be the hepatic artery. Now, mainly hepatic artery carries oxygenated blood from the heart itself together with glucose that supplies all this to the liver so that it can actually continue to respire and have energy for its metabolic function. Now, there are two veins that are connected towards the liver. The vein that's going out of the liver will be known as the hepatic vein. And in contrast with the hepatic artery, there will be much lower O2, much lower glucose, as well as an elevated carbon dioxide as the result of the waste of aerobic respiration. Now, however, the most important vein is the one that actually connects the intestine towards the liver itself. So labeling over here, this will be considered to be your hepatic portal vein that actually sends the digested material in the small intestine to the liver itself for some post-processing. Now, some of the items here we're looking at will involve the conversion of excess glucose to glycogen, the act of deamination of excess amino acid to urea, detoxification, mainly from the act of breaking down your alcohol, facilitated by enzymes such as alcohol dehydrogenase, as well as the breaking down of red blood cells and collection of iron that is found in the hemoglobin itself. And lastly, liver has a very important digestion role, especially for lipid, which is the production of bile. So bile itself is actually produced by the entire liver, but lipids itself will be found in the small intestine. So this is where we will employ the bile duct over here, which in this case here is labeled by X, that will actually send the bile into the small intestine to facilitate the process of emulsification. Now, emulsification does not chemically break down your lipids. However, it does change larger lipid droplets into much smaller lipid droplet. And this much smaller lipid droplet actually has a much higher surface area to volume ratio that facilitate the enzymatic reactions brought to you by pancreatic lipase or intestinal lipase, making fat digestion more efficient. So if you were to have a blockage at tube X, what will happen here is that the fat digestion will be inefficient. It will still continue, but it will be inefficient because instead of digesting your lipid droplet, we will actually be digesting lipid itself, which have a much lower surface area to volume ratio. Now, urea formation will not stop because urea is not sent to the liver via duct X, but rather through the hepatic portal vein. Same for glycogen and same for the detoxification of alcohol, all which is courtesy of the transportation via the hepatic portal vein from the small intestine to the liver. So A is the answer here.